Hello class, this lecture is going to be on the chemical control of microbes, and you have this chart available for you on Adverum. So my suggestion is to kind of follow along and take some notes with me as we go through all these different types of methods to destroy microbes. So the first part I'm going to go over is a little bit of vocabulary for you. The difference between disinfection, sterilization, antisepsis, and sanitization. So disinfection, I'm sure you've done this in your own home when you're trying to disinfect your bathroom or your kitchen and you spray some chemicals and wipe it down. So what is the goal here? The goal is to try to get rid of the vegetative pathogens. So what does vegetative mean? I'm going to use the term alive for vegetative. So vegetative means the microbe is doing all of the normal life functions. It is growing, it is dividing and reproducing, it's metabolizing, using energy, using nutrients. It's alive. Now, the opposite of that would be that it can be dormant. So some microbes, such as bacteria that we talked about, especially these endospores, can go into dormant stage, meaning that you've changed the environment in some way that's unhospitable, and they don't like that, so they're going to be dormant until it returns to a situation where they're happy. So it returns to the temperature that they want or the nutrients that they want, and then they go back to becoming vegetative again. The other main point here for disinfection is that this is on inanimate objects. So you can use quite harsh chemicals when you're spraying inanimate objects versus over here with antisepsis, we're going to put that on body surfaces. And sometimes what you'll see is it's the same chemical, okay? So you might use the same chemical, but you might have a much lower concentration when it's used as an antiseptic because you're rubbing it on someone's skin. So maybe use a little bit of alcohol or a little bit of iodine on their skin before you cut into it during surgery or before you poke them with a needle. But with disinfection, you can have much higher concentrations. And then, of course, some tools are going to be more prone to disintegration with chemicals. So things like plastics, you have to be more careful versus metals and glass that are much hardier under various types of chemicals. So how is this different from sterilization? In sterilization, you're not just removing some pathogens. So in disinfection, it's, let's say, some to most that you're trying to remove. But in sterilization, you should be removing all, okay? And so sterilization is much more difficult to achieve. Often it takes longer, it takes higher concentrations of these chemicals, but in some situations we really need that. So for example, if you're going into something like surgery or you're going into dealing with certain populations such as burn victims who are much more prone to getting infections, then the sterilization is of vital importance versus people in their own homes. They're just disinfecting their bathroom and their kitchenware. They're not as concerned about sterilizing it. And then notice decontamination, sanitization. There's some overlap here with the disinfection, but the main idea here is it's mechanical removal. So one thing that's really interesting when you guys are designing your hand washing experiment, for example, is that it's not just about the chemical that you use, it's also about the technique that you use. So for example, how are you rubbing your hands together? What are the different motions that you're using? Are you scrubbing hard enough? Things like that. So mechanical removal of the microbes and then washing them off by rubbing with a wet towel or a sponge can definitely make a big difference there. Now, some other vocabulary that you want to be aware of is whether a term ends in cidal versus static. Okay, so what do I mean by that? So potentially you might have a bacteriostatic material or chemical versus a bactericidal, right? So bactericidal means you are killing it, you're killing it to the point of no return. So if it dies, then it can never come back again. Versus static, you're just inhibiting the growth. Okay, so you're inhibiting the growth, meaning if that microbe, in this case that bacteria, is put back in an environment that has the nutrients and the temperature that it wants, then it can go back to being vegetative again. So inhibiting growth, examples such as, think of like your refrigerator, refrigerating something, or even freezing something. You didn't necessarily kill that microbe. If you put something in the refrigerator and it's frozen, and then you removed it and put it in a sterile environment, like in a new sterile room, and allowed it to thaw, there would still be microbes in there that can become active again. So you haven't actually killed them, versus putting them in a situation where it's cytal and then you're actually killing them. Now, if you go back to the chart that we saw where it's all together, there are 3 different categories here. We've got our physical agents, our chemical agents, and our mechanical agents. And you can sometimes use, a combination of these. It's not necessarily that you only use one of them, but this is just one way to organize the material that your book chose to use. So let's start with the mechanical removal, since that's the smallest one. So let's take a look at what's going on here. Filtration usually has to do with the size of the pores in the material and in the filter. Okay, in the filter. So the smaller the pores are in the filter, the more microbes it can remove. And usually the size is something like 0.02 to 8 microns. And obviously the smaller the pores and the better your filter, the more expensive it is as well. So you kind of have to look at the situation. Why are you using this filter? What do you need it for? Air filters, for example. The hospital uses something called a HEPA filter. And the HEPA filter has a 0.3 micrometer size pore. So this is, it's actually called high efficiency particulate air filter. So clearly, hospital is going to need a better filter than maybe somebody who's just putting a filter in their air conditioner or a filter in their vacuum cleaner or whatever. Those filters aren't going to have this restriction. Then we've got here sterilizing certain liquids. So we'll talk about sterilization methods of liquids such as pasteurization, so heat, various types of chemicals you can use. Think of like chlorine in your water. The problem is sometimes chemicals and heat are not allowed, okay? So if we can't use heat or chemicals, then what's left, we can use filtration. So examples of things that would use that would be blood products. Okay, so you can't heat the blood, that's going to destroy the cells. You can't use chemicals on the blood because you're going to put it inside of somebody's body. You can't do that. We also have this problem with vaccines. So anything that you're injecting into somebody, you have to use this filtration method is the best way to remove potential microbes. All right, so that takes care of our mechanical removal. Now let's continue on with physical agents. So the first part here is going to be heat. Heat is an excellent way of destroying microbes. What it does for the most part is it's going to denature proteins. Okay, so even something like boiling could denature the proteins and change their shape to a point where those enzymes, those vital enzymes for life function are no longer going to work, and then the microbe is going to die. Then you also have like super high heat. Super high heat doesn't just denature proteins, it completely incinerates the organism. So it is burned to ash, it is burned to a crisp. There is nothing left of the cells in that case. So what are some different types of heat? We just said incineration right there. So incineration is what we're going to use for sterilizing. Okay, and it's going to be super hot. So think of the Bunsen burners that we used in lab. Okay, so the Bunsen flame. Bunsen flame uses a gas, so it's not the same as like a candle flame. If you've ever burned yourself with a candle flame, I mean, it's already pretty hot. But we're talking about Bunsen flame being something like thousands of degrees Celsius. And you can tell because the little metal loop that you put in there turns red hot. So that is a very high temperature, and if you do it nice and slow, turns red hot, it is going to incinerate any type of microbe that was on that loop, and it will be sterile. Of course, after you remove the loop, so I will just write inoculation loop. Inoculation loop was the example. Once you remove the loop, if you set it down on the table or something like that, it's going to get contaminated again, so you have to make sure you use it immediately. You can also use this technique for sterilizing, all sorts of metals can handle this, and then maybe glass can handle really high heats, though not necessarily to the same level as the metals. Okay, so what about a dry oven? A dry oven can also be used to incinerate. Oh, by the way, actually, let me just add one more thing. There's something called an autoclave. Okay, so an autoclave is a device that can be used to sterilize, your metal tools. And if you had glass petri dishes, you could use an autoclave for that. We did not have one in lab, which is why we would throw our plastic petri dishes out into the, to the hazardous waste. If they had been made out of glass, and if we had an autoclave, we could have actually reused our petri dishes. So going back to this, the dry oven, it's a much lower temperature, but it's still super hot. So you're looking at 150 degrees Celsius, but because it's not quite as high of a heat as the incinerator, you're looking at waiting between 2 to 4 hours, at this temperature to sterilize something. So this is, of course, this was our dry heat, right? These are both types of dry heat. So now let's look at some moist heat. Moist means there's some kind of liquid involved, right? So you've got steam under pressure. And this also can be done in an autoclave. Okay, so you can have an autoclave with steam under pressure. You've got 121 degrees Celsius, which is still super hot. You're waiting about 20 minutes at that temperature for this to work. This is great for your petri dishes. It's great for your surgical tools. And that the exact protocols are going to differ depending on the scenario and what kind of tool you're looking at. I think I mentioned earlier that things like prions. Prions are super resistant to things, as well as endospores. So something that would destroy your typical bacteria or your typical virus might not be hot enough, or you might not be waiting long enough to destroy the endospore. So if you're concerned about endospores, you might have to increase the amount of time or the temperature. With prions, it's really, really bad. So you'd have to find a whole different protocol for destroying potential tools that have been infected with prions. In some cases, they're just going to throw them out and not even bother trying to sterilize them again. What else do I want to say about steam? There's also something called ultra-high steam. So kind of running out of space here, but ultra-high steam is going to be 134 degrees Celsius instead, and you can do that just for a few seconds. So this is something that they do with milk, which is kind of interesting. Rather than just pasteurizing it normally, they can use this method to sterilize milk. Okay, so now let's look at disinfection. Disinfection, again, if you recall, so now we're just disinfecting. We're no longer sterilizing. We're trying to get rid of as many microbes as possible, but some of them might survive, such as those endospores. So boiling water, this is kind of the recommendation they give you if you're ever out camping or stuck on a deserted island and you want to drink some water. Don't drink that water straight out of the stream because it's going to have all sorts of microbes in it. So you can actually boil it, and when you're talking about boiling, that's only like around 100 degrees Celsius. So clearly not as hot as this steam under pressure situation we were just talking about. This is also where pasteurization falls. So regular pasteurization, we do boil it. You can actually flash boil it faster than not just putting it in a pot and waiting for it to boil really slowly, but you can flash it. Flash pasteurization only takes about 15 seconds. And then you can also sometimes do it slower than that. So that's your moist heat. Okay, so let's move on now to radiation. So what's going on with radiation? With radiation, you're using really high energy beams. So let me put that here. So high energy beams. So what does that mean? So not regular light. Okay, regular light is something that is visible to us, right? Visible to our eyes. And the wavelengths of that type of light is not considered very harmful. Electromagnetic waves that are even less energetic than light would be something like radio waves. What else? So yeah, anyway. You can look it up, but regular light is not going to be harmful. So we're going to look at higher energy such as UV light, and then you can get even higher energy than that, such as gamma waves. So gamma waves are something that exist in space. It's something that you have to worry about if you go out into space. So our astronauts have to worry about that. If they're doing a spacewalk, that's one of the reasons they have that spacesuit on. It has to protect them against this kind of radiation. But here on Earth, we're protected because of our atmosphere. So microbes don't like this kind of really high energy radiation either. And even UV light. So UV light can be used to disinfect. Again, this is disinfection, not sterilization. And what we do is, this is not uncommon for certain food items, such as like berries. Berries where you can't really scrub or rub them really hard with a wash. You don't want to have a bunch of chemicals on them. So you could use UV light to irradiate. It's called irradiation, actually. Irradiation of your berries, and then your blueberries, strawberries, and so forth will last longer. So you could look up a picture of irradiated fruit versus not irradiated, and you will see that the ones that don't undergo this process will develop mold and other problems a lot sooner. So they're going to start to rot. The wavelength for this is 260 nanometers. I don't really expect you to memorize that or anything, but that means it's at a much higher energy than our regular light. So what's actually happening, though, it's kind of cool, is it causes mistakes to happen in DNA. So DNA should have very specific kind of bonding rules, and what happens with UV light is it causes these special types of bonds called, let me see if I can spell it right here. So pyrimidine dimers. Here we go. So pyrimidine dimers, and you can look up a picture in your book of what they're talking about, but it's just this abnormal bond inside of the DNA that causes mutations, and eventually it causes that microbe to die because it becomes non-functional. So again, this is just disinfection. You didn't necessarily destroy everything. So what could you do instead? You could use gamma rays or X-rays. Both of those are much higher energy beams. And just like X-rays and gamma rays are harmful to human cells, they're going to be harmful to our microbes as well. And what they do is they denature proteins again. Okay, so they're affecting their proteins, and then they're also going to affect their DNA, but they're going to affect their DNA in a much worse manner than the UV light did. So instead of just causing a few abnormal bonds here and there, it's going to break up the DNA into non-functional units. All right, so that leaves our middle section, the chemical agents. And so the problem with this part is that a lot of it is actually the same chemicals over and over again. It just kind of depends on the concentration. So for example, antiseptics. What are common antiseptics that are considered okay to use on the skin? You've got your iodine that kind of has this like brownish tint to it. You've got alcohol. So think of your alcohol gels that people are using right now, they're hand sanitizers. You've got hydrogen peroxide, but at a pretty low concentration, only 3%. So 3% hydrogen peroxide is sort of your standard over-the-counter hydrogen peroxide you can buy at the store. It does sting and fizzes if you put it in a wound. You definitely shouldn't be drinking it, but it's safe enough that, you know, it's considered safe for over-the-counter everyday use. You can actually rinse out your mouth with it as a mouthwash because it doesn't sting that bad. Then we have some other things, various types of dyes, soaps. You can look up more detail in your book, but different kinds of dyes and soaps, and actually some types of metals such as mercury or silver, but again at a low concentration. So then what happens when you move over to disinfection? So antiseptics was on live, you know, like skin or wounds. But disinfection and sterilization is going to be on inanimate objects. So things that are not alive, such as your tools, tables, surgical equipment, and so forth. So you can use a much higher concentration. Things you're going to see here include chlorine, which can also be used in water too. So actually, well, I don't think there's anywhere really to write that, but chlorine, of course, is used in pools. But we can have a much higher concentration of chlorine and then just kind of dump some tools into it and leave it there for a couple hours while it disinfects. We also have various types of phenols, which is a kind of chemical. They basically disrupt the proteins and the membrane of the cells. We can have alcohol, but at a higher concentration. These guys kind of dehydrate the cell, so that's kind of a cool thing that they do, is they dehydrate the cell as well as disintegrate the proteins. H2O2, but at a higher concentration, we're looking more at 6% and above. We can use soap again, but you can use stronger soaps. Think of like the detergent that you use in your laundry. If you've ever gotten that on your skin, you know that it's kind of uncomfortable, and by the time you wash it off, your skin is kind of like red and raw if you leave it on too long. We can use formaldehyde. And you may know formaldehyde is pretty nasty. You don't want to be exposed to it too long because it can actually cause cancer. You also don't want to be breathing it in. It can evaporate and become gaseous quite quickly. Types of acids can be used for disinfection. And then think of something like bleach. Okay, so bleach is a very strong antimicrobial. It's one of the few things that will still kill endospores. And it's sort of like the go-to chemical in hospitals to kill C. diff. Then we also have something called quaternary ammonia compounds. Those are various types of detergents that are used to disinfect all sorts of things in the hospital, including valves. So valves that are going to be used for transplant, think of artificial joints. And then they can just be used for basic cleaning too. It kind of depends on the concentration. It can be used as a spray to completely decontaminate a room. And it can also be used to clean catheters. Catheters are kind of a nightmare to clean, as you'll find out, because biofilms love to grow on them, and there's sort of a bridge between the outer environment and the person's inner environment. So it's just a fantastic way for microbes to enter the body, whether we're talking about urinary catheters or catheters for IVs and things like that. And then sterilization is going to be a lot of the same stuff that we just saw, but at an even higher concentration and for longer periods of time. And then over here on the gas, so the difference between gas and liquid is just sort of how are you applying it to your materials. Are you spraying it from a can? Are you actually fumigating? So that's basically the word you want to look at. I don't know how to spell it, but fumigate. You can fumigate a room. You can have these chambers that are filled with the chemical, and then they'll increase the pressure in there and basically sterilize using gas. And so some of the chemicals here that we'll see is supposed to be chlorine. So chlorine again, hydrogen peroxide. There's some other things like ethylene. And then anything else I want to add here. So some other vocabulary for you. If it's aqueous, that just means that you added water to it. Okay, so you had some kind of chemical compound and you added water to it. But if you see the term tincture, that means that you mixed it with alcohol. Okay, so some chemicals are mixed with alcohol to be used before application. All right, so I think that concludes everything I want to say about this chapter. And of course, you can read more details as well.